Assalamualaikum warahmatullahi wabarakatuh to Dr. Nick. So in this video, our group will be presenting about gene editing in stem cells, which is harnessing CRISPR for regenerative medicine and tissue engineering. So this is our group member. Firstly, me, Radina Aisha binti Saidan, and my members, Jessica Joyce Julian Henry, and also Nur Amira Afifah binti Tahir. So in this video, we'll talk about the introduction, advantages, application, limitation, and also conclusion. So now, let's move on to the introduction. So, regenerative medicine and tissue engineering have long sought the holy grail of repairing damaged tissues and organs. Recent breakthrough in gene editing with CRISPR-Cas9 have offered a transformative tool for this pursuit, especially when combined with the remarkable potential of stem cells. The research delved into the exciting merit of these two revolutionary technologies, exploring how CRISPR-mediated stem cell manipulation can pave the way for personalized therapeutic approaches, disease modeling, and also the creation of functional replacement tissue. So now I will explain about regenerative medicine. Regenerative medicine primarily aims to grow tissue or organ replacement in the lab and implant them into the body or restore the function of cells, tissue, or organs. The therapy may involve the injection of cells, transplantation of organ or tissue that are grown in vitro, or in situ induction of regeneration with bioactive molecule administered alone or secreted by implanted cell. Autologous stem cells represent an important cell source in personalized regenerative medicine due to their ability to proliferate, differentiate, and secrete cytokinins or a growth factor to reshape the local microenvironment and stimulate tissue regeneration. Different stem cells can be isolated from such sources as embryo, for example, embryonic stem cell (ESCs), bone marrow-derived mesenchymal stem cell (BMSCs), adipose tissue, which is adipose-derived stem cell (ASC), and blood, which is hematopoietic pluripotent stem cell (HSCs). Moreover, induced pluripotent stem cell can be generated by programming somatic cell, which is from fibroblasts. ESC and iPSCs can pluripotent stem cell capable of differentiating into virtually all cell lineage, including the cell types that are difficult to isolate and expand. BMSCs and ASCs can differentiate into adipocyte, chondrocyte, or osteocyte and are often used in the regeneration of bone and cartilage. HSCs can differentiate into all cell types in the hematopoietic lineage and are ideal for treating blood disorder. This autologous stem cell eliminate the need for immunosuppression and can be manipulated by chemical, biological, and even genetic approach to correct gene defects or activate pre-programmed cue to guide the cell. Next is genome editing. What is genome editing? Genome editing refers to the use of engineered nucleases to edit the genome at specified locations. The genome editing system, such as zinc finger nuclease, ZFN, transcription activator like effector base nuclease, TALEN, or clustered regularly interspaced short palindromic repeats, or we call as CRISPR or Cas9, are designed to induce DNA double-strand break at predetermined genomic loci, which can be repaired to two pathways. Without a homologous template DNA, the double-strand break is repaired through the error-prone non-homologous end joining, which directly joins broken chromosomal ends and result in the insertion or deletion. If the nucleus is designed to induce two cuts at distant genomic site, large DNA deletions can be achieved. With a template DNA carrying sequence homologi with the targeted locus, the double-strand break can be repaired by homologi directed repair pathway. Depending on the homologous sequence in the template on the DNA, the homologous directed repair pathway allows one to precisely integrate, delete, or edit DNA sequence at predetermined sites. This method is similar to the traditional homologous recombination, yet the nucleus acid double-strand break significantly improve the homologous repair efficiency by several order of magnitude.
What you're seeing here is a key moment for the gene editing tool CRISPR-Cas9. That's because the doctors are injecting a patient with an experimental drug that will edit their DNA inside their body. CRISPR-Cas9 holds a lot of promise, but up till now, all the treatments in human clinical trials have been ex vivo. They have relied on taking cells out of the body in order to edit them. Like in the treatment for sickle cell anemia, researchers removed cells from the bone marrow of a young woman in Mississippi, modified them to turn on the production of fetal hemoglobin, and then infused the cells back in. But for many inherited diseases, like brain, muscle, or kidney disease, you just don't have cells that you can take out and put back in, which is why the first in vivo trial, called Brilliance, is so important. So let's take a look at how the in vivo gene editing drug works. CRISPR-Cas9 technology is adapted from bacteria and archaea, specifically from a defense system used to detect and destroy phages. And it's really appealing to researchers because it's cheap, precise, and highly adaptable. It's also elegant.
Its basic principles are fairly straightforward, although there are many modifications of this and many layers of additional technologies now being deployed. But at its core, it's a combination of a Cas9 enzyme, which is really good at cutting DNA, and a guide RNA, which directs the Cas9 enzyme to a specific location in the genome.
This guide RNA is designed to be unique to each application. It contains a 20 base pair sequence called a spacer region that's designed to be a homologous base pair match to the cell's DNA. As this whole complex bounces around the cell, the Cas9 attaches to the cell's double helix at short sequences called PAM. The specific PAM sequence depends on the Cas enzyme being used. Each time, this unwinds the double helix just a little bit, allowing the guide RNA to check if there's a match. If not, the complex moves off. But if those base pairs do match, the guide RNA will continue to anneal to the target DNA. Once the Cas9 detects that every base pair of the spacer region is a match to the cell's DNA, it cleaves the double helix. And this by itself can be enough to knock out the gene. Scientists can also provide a repair template to the cell. This is the basic way the technology works, but CRISPR-Cas9 systems are being rapidly developed, so it can also do lots more. Now let's take a look at a CRISPR-Cas9 treatment in practice. The in vivo trial I mentioned earlier is testing a drug for Leber congenital amaurosis type 10, a disease that affects photoreceptor cells in the retina and causes severe early onset vision loss. To get the drug into the eye, the doctors performed a subretinal injection.
The surgeon makes 3 tiny incisions in the eye so they can put instruments inside the eye to remove the vitreous gel, which is needed so that the very fine cannula that's used to make the subretinal injection can be passed across the eye, placed very delicately under the retina to inject this small amount of gene editing drug.
That drug contains an adeno-associated virus vector. The virus is the delivery system for CRISPR-Cas9. So the researchers put the sequence encoding the Cas9 protein and 2 guide RNAs inside the virus, and the virus carries it like cargo to the cells. The target is a mutation in photoreceptor cells, in the CEP290 gene.
So the most common mutation in the CEP290 gene that's associated with severe early onset vision loss is an intronic mutation that results in the inclusion of a cryptic exon, an abnormal exon in the gene, so the gene doesn't function.
Introns are the non-coding parts of sequences that get removed during RNA splicing. Exons get left in. And that one wrong letter means that not all of the intron gets removed.
So it's a great target for an early CRISPR therapeutic because all we need to do is cut out mutation in the intron.
And that, the researchers hope, will restore gene function to the cells. They're looking to enroll 18 patients, with results starting to trickle in 2021. But while they're going to be paying close attention to the efficacy of the medicine, the primary outcome is safety. And a lot of uncertainty still remains about CRISPR-Cas systems more generally. But one thing is clear. We are finally at a point where the promise of CRISPR-Cas9 is beginning to be tested out in the real world.
Imagine a world where genetic diseases are eradicated at their source, damaged tissues seamlessly regenerate, and personalized medicine become a reality. This is the transformative potential of gene editing in stem cells, a technology poised to revolutionize regenerative medicines in a new era of healthcare. Okay, so the first advantages is engineering cells for tissue regeneration, where CRISPR isn't just about fixing broken genes, it's also about building a new life. This innovative technology acts as a skill architect, where it guides stem cells to become specific cell types needed for repair. Imagine turning a blank slate into a specialized builder, ready to reconstruct damaged tissues from bone and muscle to skin and even vital organs. But the possibility doesn't stop there. CRISPR also allows us to create designer stem cells with enhanced properties for tissue replacement. Just like customizing a race car for peak performance, we can also engineer stem cells to be stronger, more resilient, and even resistant to diseases, ensuring optimal success in tissue regeneration. The ultimate goal for CRISPR is to grow functional tissues in the lab for transplantation, which eliminates the need for donor organs, reduce rejection risk, and holds immense potential for treating a wide range of conditions. Like, can you imagine growing a new heart or kidney right in the lab, ready to be transplanted and restore someone's quality? Next is boosting cell function and survival, where CRISPR makes cells thrive. This technology can enhance the therapeutic potential of stem cells by improving their survival and engraftment after transplantation. Think of it as giving stem cells a superpower boost, ensuring they reach their destination and successfully complete their repair mission. But the benefit extends beyond mere survival. CRISPR allows us to develop stem cells resistant to disease or stress. Imagine soldiers impervious to enemy attacks. These resilient stem cells can withstand harsh environments and perform their tasks even in a challenging condition, and maximizing their therapeutic efficacy. The third one is correcting genetic defects, where we no longer have to settle for treating symptoms, because CRISPR can empower us to fix the root cause of genetic diseases at the DNA level, which offers hope for previous untreatable conditions like cystic fibrosis and sickle cell anemia, providing potential cures where none existed before. Furthermore, CRISPRs also unlock the door to personalized treatment options. By targeting specific genes, we can tailor therapies to individual patients, accounting for their unique genetic makeup and disease progression. This paves the way for more effective and efficient treatments with fewer side effects. And last but not least, the advanced therapeutic applications of CRISPR in stem cell therapy are vast and far-reaching. This technology holds the potential to treat complex genetic disorders that we were once considered untouchable, offering hope to millions who previously had none. Age-related diseases, neurodegenerative conditions, and even cancer No challenge is too great for CRISPR's transformative power. Furthermore, CRISPR can improve existing stem cells therapies by optimizing cell delivery, reducing off-target effects, and minimizing immune responses. This translates to better outcomes for patients, faster recoveries, and a brighter future for regenerative medicines. And finally, CRISPR opens the door to tailoring treatments to individual patient needs. Forget about one-size-fits-all approaches. This technology allows us to create personalized therapies that take into account a patient's unique genetic makeup and disease progression. This precision medicine approach promises more effective and targeted treatments to maximize the benefits for each individual.
Advanced glycation end product may be a treatment strategy through anti-inflammatory effect by transfection of Cas9 protein, sgRNA, and the donor plasmid encoding the sRAG gene into BMSCs. There is the experiment about the sRAG gene was integrated into AAV as one site and generate the sRAG secreting BMSC. Implantation of the sRAG secreting BMSC into corpus luteum of Parkinson's disease mouse models and inhibition of RAG in neuronal cells significantly reduced the neuronal cell death in corpus luteum and substantia nigra. Next is back pain is leading cause of disability worldwide as is linked to degenerative intervertebral disc (IVD). Degenerative IVD triggered interleukin 6 induced increase in neuron activity to thermal stimuli, which is directly mediated by AKAP, which is a kinase anchoring proteins, and enhanced by acidic pH. In light of this, there is the study about LV delivery of Cas9-KRAV and sgRNA were employed to knock down AKAP in red dorsal root ganglion neurons, which abolish degenerative IVD-induced elevated nociceptive neuron activity to the thermal stimuli. The study suggests the feasibility of CRISPR-mediated knockdown in nociceptive neuron as a possible neuromodulation strategy for this dysgenic pain. Peripheral nerve injuries may lead to disability and remain a challenge in the clinical setting because more than 50% of patients do not fully recover after treatment. To promote peripheral nerve regeneration, the researcher constructed a hybrid BV vector to carry the SAM-based CRISPR-A system, including TCAS9, PP64, MPH, and also a sgRNA array, which targeting 9 positions of the neurotrophic genes, which are BDNF, GDNF, and also NGF. To exploit the log speed-based hybrid BV system, the entire cassette, around 12.7 kilobase, was flanked by 2 log speed sites. Post-transaction of red ASC with the hybrid BV and another BV expressing pre-recombinance leads to persistent and simultaneous overexpression of BDNF, GDNF, and NGF for at least 21 days. Furthermore, the BV vector was able to transduce ASC cell sheet effectively, thus allowing them to functionalize ASC cell sheet to produce the 3 neurotrophic factor. Implantation of functionalized ASC cell sheet to the sciatic nerve injury site in rats substantially promote nerve regeneration in vivo, thus it underscoring the potential of CRISPR-A system in nerve regeneration. So overall, CRISPR or Cas9 show potential for treating neurodegenerative disease like Parkinson's disease by gene editing or protein delivery, modulating pain pathway through CRISPR-I in pain signaling neurons, and lastly, regulating neuronal activity and excitatory inhibitory balance in the brain.
Alright, so now I will be explaining on the second application of CRISPR technology in preventing muscle degeneration, which focusing on 2 major types. First is Duchenne muscular dystrophy, DMD, and second myotonic dystrophy type 1, DM1. Alright, so let's start with DMD first. This is a disease that caused by mutations in the DMD gene, leading to progressive muscle weakness and wasting. CRISPR-Cas9 delivered by adeno-associated viral AAV vectors can delete specific exons in the mutated DMD gene and restore functional dystrophin expression. This approach has successfully corrected the gene defect in mouse and dog models, demonstrating its potential for DMD treatment. CRISPR-A, a gene activation technique, can also be used to stimulate utrophin, which is a protein with similar function to dystrophin, and to offer an alternative therapeutic avenue. Ex vivo editing of patient-derived muscle progenitor cells and induced pluripotent stem cells, iPSCs, allow for personalized treatment options tailored to individual mutations. Knock-in strategies in iPSCs can hold promise for introducing functional full-length DMD gene copies, offering a potentially curative approach. Right, for the second one, which is myotonic dystrophy type 1, is a disease caused by expanded CTG repeats in the DMPK gene, which leading to muscle weakness and other symptoms. And a dual sgRNA strategy using CRISPR-Cas9 can efficiently excise these expanded repeats in the DM1 patient-derived iPSCs and myogenic cells. This approach offers a potential gene correction method for DM1, potentially halting disease progression. So next, I will pass to Jessica.
Next, application of CRISPR in bone tissue engineering involves the immortalizations of bone marrow-derived mesenchymal stem cells, as known as BMSCs. Utilizing CRISPR, the SV40T oncogene is integrated into BMSCs, allowing for their long-term culture. This immortalization process lays the foundation for further genetic modifications. For instance, adenoviral-mediated expressions of BMP9 in these immortalized BMSCs not only promotes osteogenic differentiation in vitro, but also facilitates bone formation in vivo, showcasing the versatility of CRISPR in advancing bone regenerative strategies. In the realm of calvarial bone repair, CRISPR plays a crucial role in stimulating osteogenesis and suppressing adipogenesis. Specifically, CRISPR activation, as known as CRISPR-A, of Wnt-B in rat BMSCs has been employed to enhance osteogenic differentiation. Furthermore, the co-activation of Wnt-B and FOX2 using CRISPR-A not only further intensifies osteogenic differentiation, but also contributes to provide in vivo bone repair. Additionally, the CRISPR interference system simultaneously activating SOX9 for chondrogenesis while inhibiting PPAR gamma for adipogenesis has demonstrated efficacy in promoting cartilage formation and calvarial bone healing. This dual programming approach exemplifies the precision and versatility of CRISPR in orchestrating multiple genetic factors for enhanced bone repair. In summary, CRISPR emerged as a powerful and versatile tool for BMSC engineering in the context of bone tissue engineering. The immortalizations of BMSCs enable prolonged culture and facilitate a subsequent genetic modification. CRISPR-A allow for the precise activation or inhibition of gene governing osteogenesis, chondrogenesis, and adipogenesis, providing a nuanced approach to bone generation. Moreover, the promising result of dual programming with CRISPR interference system, manipulating multiple genetic factors simultaneously, hold significant potential for advanced bone repair and regeneration strategies. This research underscores the substantial impact of CRISPR in developing more effective cell-based approach for bone repair. Next, I'll be presenting the limitation in CRISPR technology. First of all, is the off-target effects. Cas9 and its variants may induce unintended mutations at off-target site, raising safety concerns. So, the strategy for minimizing this effect include controlling Cas9 expression using tissue-specific promoters, split or inactivate Cas9 design activated by other chemical inducers, or light and sgRNA modification like truncations and engineering hairpin secondary structures. Second is the immune responses. Cas9 protein and sgRNA can trigger immune responses, limiting in vivo applications. So the pre-existing antibodies and T cells against Cas9 variants hinder delivery and editing efficiency. And the potential solution involves targeting immune-privileged tissue for ex vivo editing, using Cas9 autologs with lower immunogenicity and modifying sgRNA to reduce innate immune activations. The third one will be delivery challenges. Efficient and safe delivery of CRISPR components is a major obstacle. Non-viral metals like R and P are less efficient than viral vectors. Potential solutions include combination delivery with dual or triple AAV, exploring alternative vectors like BV with magnetic nanoparticles, and continue research on merging vectors. Number 4 is low genome editing efficiency. Primary cells such as human PSCs and HSCs exhibit lower editing efficiency due to TP53 activation and apoptosis, as well as challenges in quiescent cell. So the strategy is to enhance efficiency include transient BCL-XL transfection for boosting iPSC survival and HDR efficiency. TP53 inhibitions and alternative approaches like homology-independent targeted integration for quiescent cell. So, as overall, we can know that while challenges persist, ongoing research focuses on optimizing existing methods and exploring novel approaches to overcome limitations in CRISPR-based technologies. So, by combining different strategies shows promise for advancing CRISPR's applications in stem cell engineering, regenerative medicine, and therapeutic interventions.
As a conclusion, we have seen that gene editing in stem cells is not just a scientific breakthrough. It is also a beacon of hope for a healthier future. From correcting genetic defects to engineering tissues for regeneration, this powerful technology has the potential to revolutionize medicines and improve the lives of millions. As we continue to explore its possibilities, one thing is certain: the future of healthcare is getting more personalized, precise, and hopeful, thanks to the power of CRISPR and stem cells. So that's all from us. Thank you for watching. Bye!